Hey everyone, welcome back. Today we are taking a quick break from our main progression and we are going for a practical pro tip episode. We are going to tackle the number one mistake that I see people make when they start working with real world and massive biological data sets. So when you transition from learning to doing, your FASTA files go from a few kilobytes to tens of gigabytes or even more. And if you open a 30 gigabyte human genome file the wrong way, your computer will run out of RAM. It will freeze and it will kill your Python script. So today I'm going to show you exactly why that happens and we will code the solution together. To demonstrate this, we will need a large file, and I don't want you to download a big file like 5 GB or something to download for this tutorial. So we will just write a quick script to generate a scale model of a large multi-FASTA file right here. Okay, so for that we will run this code and finished. So when we run this, it will take just few seconds like right now, and we have just created a file with a 500,000 sequences, and if it's about 50 megabytes. I don't know if we can see it. Yes, we can see it. So it's like this. A very simple example of a FASTA file, and we have a lot of sequences here, as you can see. Right. So yeah, not huge, but big enough to prove our point about the memory. So the pitfall. Now let's look at the bad way to read this. So in standard Python, beginners will often use the read lines function or read function. So let's use that, and we will use Python's sys module to see how much memory that resulting variable takes up. So for that, we will import sys, just a print statement, into memory. Yeah, that should do. After that, we will do with open, and our name is file name is bigtummy.fasta, and we will open it in the read mode. So R as file, okay, and all lines is equal to file dot read lines, right? And then we will check how much memory has been used is equal to dot get size of all lines divided by how much 1024 into 1024, right? Then we will print length of all lines, and we need to add an f in here, and we'll copy this line, paste it here, and here we will print memory used by the list variable, and here we will print memory used, okay, and we will, okay, I think that should calculate it into MB, right? So we will write MB over here. Okay, now let's try to run this and see what happens. Look here, just the list structure itself is eating up several megabytes of RAM, and string data inside it takes up even more. Now imagine if this was a 30 GB file. So all lines would try to be consumed 30 GB of RAM instantly. This function in particular. And if you have only like 16 GB of RAM, your computer will basically choke and the script will die. So what is the solution? The solution is line by line. So how do we process a massive file? The secret is basically one piece at a time. We don't need the last sequence in memory while we are calculating the GC content of the first sequence, right? So we can iterate through the file line by line, with the with open statement in Python, and that actually does this natively if you use a simple for loop. So we will use some code again here. Here we are going to do processing line by line. Right. Now what we will do is we will assign a line count as 0. Then now we will not use read lines here. We are going to use a different approach, as I mentioned. Big dummy.fasta read format as file. Now what we will do here is for line in file, line count and increment it by 1. Now what we will do is we will calculate the memory used is equal to sys dot get size of line, and we will use the same code again. Total lines processed this time. And here we will print line count, and I think the yeah line variable to be exact, right? Memory used by the line variable, and it will be in I think bytes this time, but we can convert it into MB as well. So we will do just that, and we'll see what happens. Okay, we did a mistake in here. Let me just move it up here and see what happens. And it's practically 0. So we will have to remove it so that we can actually see it in bytes. And there we have it. See the difference? We processed the exact same million lines, but the memory footprint was just a few bytes as compared to the full memory size. So as soon as the loop restarts, Python throws the old line in the trash and grabs the next one, and your RAM stays perfectly flat. Now we are usually don't want to parse raw lines, we want sequence objects, right? So fortunately, BioPython is built by professionals who know about this memory trap. So they designed a sequence io.parse function to be an iterator. So we will see how professionals do it. So from bio, we will import uh, sequence io. We will also import sys. Okay, my spelling is wrong. After importing that, we are going to use the same statement, just change it a little bit, and processing with BioPython this time, right? And total sequences is 0, and what else? Yeah, total basis is also 0 for the time being. Then what we will do is for record in sequence io dot parse. Now we will enter the file name here, bigtummy.pasta, and we will read it as a Fasta file, right? Now what happens inside is total sequences will be incremented by 1, and total basis will also be increased by the length of record dot sequence, right? Now let's calculate the memory used. We will use the same code. Memory used is equal to sys dot get size of, and here we will put record, right? I believe it will be small as well, so better not to convert it into megabytes. Then what we will do here is total sequences is equal to, and here we will print total sequences, right? Similarly, we will print total bases as well. Let's just copy and paste and save our time. Total bases, and here we will use total bases. And for the memory, same as this one, but memory used by the, what are we using? Current record, right? Now let's see what happens. And here we have it. See, boom. We just analyzed half of a million sequences, counted 25 million bases, and at the end of it, the record variable in our RAM is still just a few bytes, and it's even much smaller than the previous one. Because of this, you can process a file of infinite size on a cheap laptop, basically, but not for surely. Yeah, so let's recap the golden rule of bioinformatics coding. Never use dot read or dot read lines on biological datasets. Always process them iteratively. So I hope this live demo save you hours of frustration and crashed computers. It's a rite of passage to kill a script this way, but now you know how to avoid it. So yeah, we will see you in the next video. Thank you.